and I'll hand you over to my colleague Pippa.
Hi everyone. So we've been on holiday, so we've had a little bit of rest over the summer holiday. So welcome back to, as Finn said, to the UK Freshwater Quality Research Programme webinar series, where we hold a webinar each a month. For those new to the webinar, welcome. And just got the objectives of the programme here for you to read. It's a research programme that's funded by Natural Environment Research Council and DEFRA. And at the University of Leeds, we oversee the programme. And as part of that, we have this monthly webinar series, which has been going for almost a year now. So the programme is made up of 5 projects. So Finn, if you could, yeah, thank you. So as I said, at Leeds, we're the champion team and we oversee that. And we've got these 5 projects, which we've heard from this year. So we're hearing from a range of other stakeholders now that are researching within the freshwater area to do with pollution. You can catch up with the previous webinars from our website. And these are the ways in which you can contact us. So as I said, you can catch up on previous webinars at the website. You can email us at freshwater@leeds.ac.uk, and you can follow us on X. And obviously there's the code there as well. But this month's webinar, I'm pleased to say, is to be given by Laura Carter from the University of Leeds, and she's going to be talking to us about co-contaminant exposure in the aquatic environment following antibiotic manufacturing. So I'm going to just pass over to Laura, and Finn's just reminded me that if you're interested in our other webinars for October and November, this is the titles of the webinar who'll be speaking. And as always, we do encourage members of the audience, if you've got an area of research that you would like to present at one of our webinars, then please do get in contact with us. We'd be happy to host you. So over to you, Laura.
Thank you, Pippa. I'll just share my screen. Okay, hopefully everyone can see that. Is that okay?
That's fine, yeah, I can see it.
Perfect. So yes, thank you, Pippa, for the introduction. So I am co-I on a NERC-funded project, which is called SELECTAR, and it's designed to look at the selection for antimicrobial resistance by antimicrobial production waste. And this project is one of 5 funded projects under a NERC program that started in 2020, and it's in collaboration with the Department of Biotechnology in India. And it's a UK-India cross-disciplinary, cross-sector collaborative program. And I'm going to be talking about our project SELECTAR today. And to begin with, I just want to acknowledge that our project is very broad, and the results that I'm going to present today have really been only possible from this larger collaborative effort across multiple teams in the UK and India. So the project's led by 2 PIs, Professor Iqbal Ahmed and Professor Alan McNally, but there's a much broader team beyond this. And our project really started with the idea of trying to understand, or better understand, chemical complexity in the environment following the release of manufacturing waste. And this was with a view to investigate whether a range of different chemical entities have the potential to select for resistance. And this is because, well, I think I'm preaching to the converted, but when we're talking about contamination in the environment, the focus shouldn't just be on antibiotics. They're not the only chemicals that are present in manufacturing discharge. There's effluents entering the environment with chemicals that have retained biological potency, but include a wide variety of different chemicals, including things such as drug precursors and solvents and so on. So really the idea behind this project was to understand the chemical complexity and how that links to the development of resistance. So briefly, our project focuses on 2 case study sites in India, and these include Derabasi in Punjab and Buradi in Rajasthan. And we conducted a year-long sampling campaign, which properly kicked off from the beginning of 2022. And we collected water, soil, and sediment samples. Now, these sites were specifically chosen because of them being known manufacturing or industrial hubs. And we had access to manufacturing effluents as well as sites in the receiving environment. So our sampling was actually delayed due to COVID, and access to our sites was much later than we anticipated. And we had to change our project design quite a bit. Like originally, I was planning to put out some composite samples, passive samplers to collect composite samples. This never materialized. We were also planning for a 2-year campaign, but because we were unable to access a number of the sites until later on in the beginning of 2022, what we've tried to do is redesign our sampling campaign to capture some spatial and temporal variability, and then complement this with some more targeted experiments in the lab. And what I'm going to focus on really today is the monitoring campaign. And the data that's been created has been possible due to complementary analysis and expertise across India and UK teams. So the chemical analysis, when we went into the field and collected the samples, was really to reveal insights into the chemical complexity. So coming back to that big picture aim that we had at the beginning of the project. And then we were trying to pull this together with information through microbial analysis to really explore which components of the production waste can impact on the diversity and the abundance of microbial communities, with the potential to select for the evolution of resistance to antibiotics. And to begin with, I just wanted to start with this summary slide from our high-resolution mass spectrometry analysis, which shows the total number of chemical entities that are detected in the samples across the different sampling points. So just to confirm, we used high-resolution mass spec to identify which chemicals were present or chemical entities were present in the samples. We didn't use this for quantification, but we have followed this analysis up using targeted analysis to later confirm chemical concentrations. But really the key take-home messages from this slide is that we were seeing quite a significant change in the number of chemical features or chemical entities that we were able to detect at the beginning of our sampling campaign and at the end. So we were seeing about 10 times greater number of chemical entities from the beginning of 2022 towards the end of our monitoring campaign in 2023. And just in comparison, some of the work which we've been doing in the UK, looking at wastewater effluents, we're typically seeing around 500 chemical entities to level 2 criteria. And in our Indian sampling campaign, we were seeing around 2,500 to 3,000 chemical entities. So what we're dealing with is quite a different environmental dynamic monitoring campaign, and it's quite interesting to think about this from the temporal aspect as well, because the difference between the number of chemical entities detected in the beginning at the end is something that we're working through at the moment, but it's something that's been mirrored in a lot of the other projects as well. And some projects have been able to include some ethnographic research alongside their chemical analysis. And it suggests that during COVID and towards the end of the lockdowns associated with COVID, there was a reduced number of manufacturing facilities that were actually working and producing the chemicals, and the ones that were operational were focused on a subset of contaminants. And the results from our data suggest that that is the case in the case study sites that we've been able to see, because we're seeing this change in the number of chemical entities. And the data analysis from sampling batch number 4, this was completed just last week, and it does confirm that I think there are about 2,500 chemical features in total in this sample. So it is like a trend that we've been able to observe in the data set. So for the moving on to the results, really, so focusing on Derabasi and Punjab first. Derabasi is located in the northwest of India, and our sampling here centered on the river Gaga, and it flows through Derabasi. So we have the Derabasi city here, and it's important to highlight that the river Gaga originates much further upstream, so in the foothills of the Himalayas. So whilst I have defined here, we've got Gaga River upstream, the drain, and the downstream sampling point, we are not defining the upstream site as being a pristine site. I think you can see from the picture, it's not pristine by any stretch, but it was a comparison to the downstream site of the waste discharge. And what's also important to point out here is that the seasonal flow of the river is dependent on rainfall. So then we're expecting dilution to play a key role when we're thinking about contaminant presence. So thinking about the frequently detected chemical classes that we're able to see, it confirms our idea really that the presence of antibiotics, they are there. They are higher in the drain that's going into the river in comparison into the receiving environment. But what we are seeing are greater numbers of non-antibiotic pharmaceuticals. So we saw quite high levels of antivirals, antihypertensives, and antifungal chemicals. But yes, we see higher numbers of chemical entities present in the drain, and typically higher numbers of chemical entities downstream in comparison to upstream. But as you can see, and as I alluded to in my previous slide, the upstream is far from what you could consider a clean or a pristine sample. What we did see were quite high levels of water disinfectant and personal care products. Personal care products, I think, based on previous research, this is what we're expecting, but the numbers of water disinfectants was a lot more than what's typically been seen. And what we were also able to see is where we had a sampling that was just after the monsoon season, this is when we saw a higher number of chemical entities as well in comparison to pre-monsoon. So just focusing on antibiotics, the frequently detected chemical classes were mostly beta-lactams, sulfonamides, cephalosporins, and fluoroquinolones. And our targeted analysis for quantification revealed some really quite high levels of fluoroquinolones. So these were pretty much consistently detected in all of our samples, mostly norfloxacin and ofloxacin, with levels typically around 100 nanogram per litre in the drain, and slightly less in the downstream samples. But if we looked across all antibiotics that we're able to quantify, it wasn't necessarily the story that you had the highest concentrations downstream of the site of the discharge, and we did see higher concentrations upstream for some of these chemicals. So I think this just goes to show that it's really important to consider multiple sources when we're considering the receiving environment. Now, moving on to the metagenomics work here. So I've presented a chart of bacterial genus abundance. And just for those of you who are not familiar with this, where we have colours. These indicate bacterial genre, and the sizes of the colours represent the percentage of the sample made up of the sequencing reads of that genus. So far more colours here are present in the drainage and the downstream sites in comparison to the upstream. So this shows that there are more diverse bacterial populations present with more different bacterial genre in the drainage and downstream sites. And to support this more, I guess, in terms of quantification, we looked at the Shannon Diversity Index, which is a measure of genus diversity within samples. And as you can see again here, we have a higher Shannon Diversity Index in the drain and downstream in comparison to the upstream. And this is further supported by bacterial load analysis, which was carried out by colleagues in India, which confirmed that the highest AMR bacterial load was obtained from samples collected from the downstream site. So this is tying together to tell quite a nice story. So we're seeing higher chemical presence, we're seeing greater bacterial diversity, and bacterial load in the drain and the downstream sites. Now, work at Birmingham was also able to identify antibiotic resistant genes that were present. And here, if we look across all sites, so upstream, the drainage and downstream, there were 28 common antibiotic resistant genes. Whereas if we look at either the drainage or the drainage and the downstream, we could see here that over half of the ARGs here were detected in these samples. So there's a clear association of ARGs with the drainage sites and downstream, which is then associated with this increase in bacterial abundance as well. Now, if we think about the 28 ARGs that are present across all three sites, there are a range of different ARGs that we were able to detect. I think the clinical relevance of some of these is questionable. There's a few ARGs have been widely detected for a while, such as SOL2. But if we focus just at the drainage and the downstream sites, we're able to see some ARGs which are potentially of greater clinical relevance, such as extended spectrum beta-lactamases. So you've got CMY59, but you've also got NDMM12 as well. So in summary, where we're up to with the work at the moment, and I should say that the analysis has all been complete, but we're still trying to, I guess, join up the dots here to complete the picture. But what we're seeing is consistently higher ARG abundance and bacterial diversity, higher bacterial load, and a greater number of chemical entities present downstream. Concentrations of antibiotics were again higher downstream more often than not, but this wasn't always the case. So now I'm quickly going to go and move on to our second sampling site, so Baradi in Rajasthan. And this is a slightly different concept that we took to the sampling campaign. So here we've got 4 sampling points, but we're not focused on a river or an environmental area in that sense. We focused our analysis on identified areas where we thought there would be hotspots of pollution. So we had samples collected at a central effluent treatment plant, or CETP, for the influent and the effluent. And we also identified a manufacturing site that discharged treated water, but we were also able to obtain samples of wastewater that was discharged from a manufacturing site into an open area that was untreated. So the take-home message from the chemical analysis is we were seeing a similar range of antibiotic chemical classes. So quite a few beta-lactams and fluoroquinolones. And again, a similar story in the sense that we were seeing more pharmaceuticals, well, non-antibiotic pharmaceuticals in the manufacturing waste and in the CETP inlet and outlet. But we did see antibiotics. There were a large number of sulfonamides and fluoroquinolones, as I said, and beta-lactams. And typically there were more of these in the CETP than in the pharmaceutical treated wastewater and in the open drain. Now, what was really interesting with this analysis was that we were able to see a large number of antivirals and antifungals, a lot more than what we were seeing at the other site. So quickly with the bacterial genus abundance, again, so we have the plot based on the colours, and what we're seeing as expected, we could see a higher bacterial diversity in the CETP samples. So this is this bottom panel here, in comparison to in the samples that were collected from the manufacturing effluent. Now, for the antibiotic resistant genes, what really stands out here is that over half of the resistant genes detected at the manufacturing site were only detected at the drainage point in the untreated water. And again, these did include clinically relevant ARGs. In comparison to the CETP, we saw a majority of the ARGs both in the inlet and the outlet. So in summary, we were able to detect clinically relevant ARGs both in the inlet and the outlet of the treatment plant and the pharmaceutical manufacturing site. At the manufacturing site, we typically saw high concentrations of antibiotics as in comparison to the CETP, and we saw higher concentrations in the untreated samples in comparison to the treated samples coming from the manufacturing site. The CETP, we saw similar types of ARGs present both in the inlet and the outlet, and this was across all of the time periods that we sampled, but antibiotic concentrations, as I said, were more variable. So to finish my talk, I really just wanted to also highlight some of the complementary work that we've been doing, both from a microbial and from a chemical point of view. And to begin with, this is a paper that Rebecca Hall led that was published recently, looking at whether non-antibiotic pharmaceuticals have inherent toxicity and whether in these long-term exposures, whether this can result in significant genetic changes with the potential or have the potential to select for cross-resistance to antibiotics. So this was building on some of the non-target screening work that we did with the high-resolution mass spec, where we were able to identify the presence of non-antibiotic pharmaceuticals. So we included here common pharmaceuticals that we're able to detect, so acetaminophen, ibuprofen, propranolol, and metformin. And also we had a treatment with titanium dioxide. And the study looked at toxicity against Listeria coli or E. coli. And this was in a 30-day selection experiment. And the results from the toxicity screen revealed that the compounds negatively impacted on the growth of E. coli. And this was across a range of concentrations. When this was further analysed through transcriptomic analysis, we identified that there were upregulated genes related to stress response and also multi-drug efflux. And this was only in response to the ibuprofen treatment, so not in response to acetaminophen or propranolol, metformin, or titanium dioxide. But then when we explored this further, we saw that compounds did not select for any significant genetic changes. And there was no evidence for selection for cross-resistance to antibiotics for the population that evolved in the presence of the ibuprofen treatment specifically. So this really suggests that, at least for these compounds that we've included in this analysis, including environmentally relevant concentrations, that there wasn't the potential to select for cross-resistance to antibiotics. in E. coli. But I think, as you can appreciate, this is a small snapshot of potential areas that we need to explore. There's a limited number of pharmaceuticals which we've done here against one strain of E. coli. So these are our findings, but I think it opens many more questions that we need to explore further. And then lastly, some of the work which we've done in Leeds has been focusing on phytoremediation of co-contaminant mixtures. And this is some of the work we're trying to wrap up at the moment. But briefly, we exposed lemna minor, so duckweed, to antibiotics, pesticides, and metals individually and both in contaminant combinations. And what we're able to see is that the presence of the co-contaminants strongly affected the uptake capacity of the individual compound. So when we're trying to think about implementing potentials for removing or treating contaminated waste, it's really important to consider this chemical complexity. So I've got, just to finish in terms of a summary, the antibiotics that we've been able to identify have the potential to cause antimicrobial resistance, alongside us being able to detect ARGs of potential clinical concern. Where CETPs are in operation and they are working, this does decrease the antibiotic concentrations for the chemicals which we were interested in, but it doesn't seem to have an effect on the number of chemical entities or the ARGs. I've talked about the importance of the contributing sources coming into the environment other than the manufacturing waste, but it does appear that seasonal trends are important for the chemical concentrations, but had little effect on bacterial genus abundance and the types of ARGs present. But as I said, this is work that's ongoing at the moment, and we're starting to try and pull out relationships between the chemical and the microbial analysis. So thank you for listening, and yes, just to acknowledge again that the collaborative effort here that's made this work possible. It's much broader than beyond my postdocs at Leeds. And I have some concluding thoughts which hopefully pull together some of the data which I've been able to present and might form some of the basis for the discussion next. But it's mostly around the need for complementary techniques, and this was based on the idea that we were seeing different chemicals present in the targeted analysis and the non-targeted analysis. Some of the pitfalls associated with grab samples when we're thinking about trying to really capture the variability and the complexity of systems. The need to focus beyond looking at just antibiotics. Specifically within antimicrobials, I think we need to increase our understanding of antifungals and antivirals. And then really looking at different scales. So I think consolidated, focused monitoring campaigns to understand processes and to better link effects are really useful to reveal fundamental insights. But we also need work at bigger scales to consider the wider ecosystem effects. So I will, leave it there. And thank you.